Hi, I'm Ted Zwicky, plant pathologist, and we developed a technique which we call the Nursery bench leachate method for detecting Phytophthora in nursery stock. This is the first of a two-part video series in which we'll be talking about this method and how it's applied in nurseries. So to provide a little context as to why we're interested in Phytophthora diseases, this is a photograph of the endangered species Arctostaphylos myrtifolia, Ione manzanita, in the foothills of Sierra Nevada, which is completely being wiped out by Phytophthora cinnamomea, a soil-borne Phytophthora species. That species has been found in multiple parts of California, causing mortality of native vegetation. It's also known as pathogen in agricultural crops, etc. And there's also other Phytophthora species that are actively causing mortality in our native plant resources as well as our agricultural and horticultural systems. The source of all of these Phytophthora, as it turns out, is the nursery plant trade, because standard nursery practices tend to allow Phytophthora to build up in nursery stock from which it can be moved out into the landscape. And this is true for both commercial nursery stock of landscape plants as well as much of the material that was produced for restoration plantings in nurseries that follow standard horticultural practice. So there's two problems with this. One, infected nursery stock is not going to survive well when planted in the landscape. And that's shown in this picture of some coast live oaks that were infected with a Phytophthora species that's not actually well characterized. It was from nursery stock directly. It's been planted out into habitat. One of the problems with planting these materials out into habitat is they don't necessarily die right away. They can keep living for a number of years, producing inoculum and spreading it into their surrounding environment. So here I have some plantings from restoration projects that are more than 10 years old. The plants are dying, but they've been in place and capable of spreading Phytophthora for an extended period of time. Now, this is an avoidable problem because if we don't use Phytophthora infested nursery stock, we won't be moving it into wildlands or other clean habitats by means of infested nursery stock. There is a Phytophthora working group noted down below here has been working on efforts to allow nurseries to produce clean stock, and one of those parts of that effort were development of best management practices, or BMPs, that nurseries can follow to minimize the chance that Phytophthora would be present in either nursery stock, effectively excluding Phytophthora from the nursery. We have a copy of these best management practices up on our website, and really they're based off of the principle that if you start clean and keep it clean, do not have the pathogen. You start clean by making sure that all the inputs don't have Phytophthora, that they're clean to begin with, and then your practices are what keep it clean, making sure that you don't introduce contamination into the nursery later on. And all these principles are really set forth in Manual 23, the Ken Baker's UC system handbook that was produced in 1957. The principles are well known, just not widely implemented. And we're trying to make it easier for people to implement these and understand what they're doing. An important part of a clean production system in which you're trying to identify and exclude potentially diseased plants from the nursery is the ability to understand whether these plants are infected to begin with. So one of the issues we have is that in many native California species of these drought-tolerant plants, we water them very frequently, and they can survive even in the presence of Phytophthora root rot. So here we have 3 toyons growing in 1-gallon containers. We can see that they're all potentially past as healthy. The one on the left here, perhaps the least healthy of the bunch. But to cut to the chase, root systems of all 3 of these plants are heavily decayed. Only the center one has some live roots in any kind of quantity, whereas the other 2 are almost have no live roots at all. So the issue is here that we can have even very severe disease and not be able to see it from top symptoms alone. Because of that, we need a really effective testing method that doesn't rely on simply looking at roots and trying to isolate from them. Because when we get into a situation where we have many plants, many of them large, many different batches, etc., we simply don't have the resources to be pulling up each of these plants and looking at the root systems and trying to determine whether the roots might be dead due to fact that they were allowed to dry out a little bit, or whether they're dead due to Phytophthora. One of the methods we can use that is simple and can be applied by nursery people is pair baiting. So we use the term baiting much in the same way as it's used if you're fishing. And the whole point in using a bait or lure in fishing is that you've got perhaps a vast amount of water, and maybe there's some fish in there, maybe there aren't. You're going to spend a lot of time if you're just going to hope to intercept one. What you really want to do is have that fish come to your bait. You want them to come to you instead of you to try to find out where they are, although you still have to be looking in the right places. And that is really analogous to what we're doing with pair baiting. So in a diverse nursery like this, we have several things that we're dealing with. We have a lot of different plants, a lot of different sized containers. They all have different propagation histories. And we're trying to determine, really, in a clean production system, are we functionally keeping these plants clean? So where would we expect to find Phytophthora, and how can we find it if it is indeed there? So baiting procedures used for Phytophthora revolve around sporangia, an asexual reproductive structure produced by these water molds. These sporangia are produced under moist conditions, and they're capable of just germinating like a spore, shown in the lower right here. When sporangia are in saturated conditions, in free water or in saturated soil, they produce motile zoospores that swim out of the sporangia and actively swim towards host tissues. This bit of video shows zoospore release occurring from sporangia of Phytophthora cactorum. You can see this sporangium here and this one are both releasing kind of in a little bit of a slow fashion. The zoospores have differentiated inside of the sporangia and are then swimming out intact. See another one releasing more rapidly down in this area. These zoospores now are swimming off in search of host tissues to which they're attracted to by chemical signals. One thing that allows Phytophthora to be a good pathogen and which can allow us to use baiting as a tool to detect it is the fact that under favorable conditions, a lot of sporangia can be formed. These are all sporangia of Phytophthora niederhauseri. You can see that they're mostly not released at this point. In this bit of video here, we're seeing what happens in a liquid solution where zoospores have been released. They seem to swim around somewhat randomly, and they do have kind of a random pattern of search, but then they home in on chemical signals that show them that there's something that they're interested in potentially colonizing. Now, this particular video is being taken from a petri plate with a little less than a centimeter's worth of water depth in it. And as we start right around here, we'll start focusing down from the top layer where we've been seeing all these zoospores swimming to deeper in the water column. As we go farther down in the water column, we'll start to see that zoospore numbers start to decrease. And as we work all the way down to the bottom where the sporangia will show up, you'll see that there's very few zoospores down there. And what's going on is that zoospores swim in opposition to gravity. Basically, they swim upward. They swim up to the top of a water column. So knowing something about the behavior of zoospores and the disease process, Phytophthora root rot in nursery plants, we're able to develop a fairly simple yet effective method to collect zoospores and concentrate them in a vessel that's baited with a green pear. We have the collection surface tied underneath this nursery bench, and it drains into a zoospore collection vessel when the plants are watered and leachate runs out from underneath these. The pear floats in that vessel, and the zoospores can be attracted to that and infect that pear. The main design parameters here is that we know zoospores swim upward, so we don't want water to overflow from the top. That's why we have a drain that comes off the bottom, so that once the water reaches this level, this upper elbow, it's going to exit through this pipe instead of overrunning the rim. We also don't pull it exactly off the bottom because there are some things in the bottom that could generate zoospores under the right conditions in the after period of incubation, which is shown in this slide when we empty out the transfer the pear and the remaining water in this vessel into a bag. The other feature we have here is that the surface area is relatively small here, and zoospores don't swim that far in solution, so we really want to keep the zoospores concentrated a short distance away from the pear bait. Phytophthora causes fairly characteristic lesions. They're a little bit variable, but once you understand what they look like, they're easy to determine, and they can be verified by means of culturing or by means of various molecular methods as well. So this video just sort of shows the process of action. The plants above have been irrigated, and the water is leaching through that. If these plants are infected, there's going to be zoospores dripping down into this vessel where the pear is floated, and it's not overrunning the rim. So we were able to come up with this concept and test prototypes and determine that, yes, this is a viable way to detect Phytophthora. But there were several other tasks. We wanted to validate this test over a variety of different types of containers and species, etc. We want to try to optimize and standardize the parameters of the test to make sure that we were doing it as effectively as we could. And once we do that, we still want to know what the ultimate test sensitivity is. What did positive results mean? What did negative results mean relative to how much infection might be in the plants? So we did a number of tests. This is one kind of more involved one, but essentially what we're looking here at is how many of the zoospores that are in the leachate are going out through the drain compared to what's maintained inside of the first vessel. And what we did find out from that is that in the standard way we initially did this, we were getting a certain amount of zoospores that were washing out the drain. We did find that by adding this flow deflector, basically a water slide, to reduce the turbulence and mixing inside the vessel, that we could reduce the number of zoospores that were just being flushed back out through the system because the water was churning so much. This slide shows another one of these types of experiments where we're taking the runoff from one vessel and running it through a second one to get an idea of how many zoospores are passing through the first one. Besides optimizing the equipment itself, we still had some other questions, and one of those are how do we irrigate these things? And how many times do we want to irrigate them? Well, we know from biology that zoospore release typically takes a certain amount of time. If we want to get zoospore release from zoospores produced in the lab, we normally chill them and then have to wait for 15 minutes to the better part of an hour sometimes to get them to release in mass. So we want to allow enough time, enough irrigations to have that happen. We also don't want to overdo it. We don't want to irrigate it longer than we're going to have released because we're just diluting the inoculum at that point. We did a number of tests where we had either arrays of infected plants, these were 1 in 15s, or just single plants that are infected. And we collected runoff from the first 2 irrigations, these are spaced 15 minutes apart, from the 3rd and 4th irrigations, or just the 5th and 6th irrigations. And in some of these tests we also baited after the fact, after the 6th irrigation. And what you see here is where we have the weakest effect is in the first 2 irrigations, and we're picking fewer zoospores up again in the post-bait treatment. Overall, our results look something like this if you're looking at the pears. And this is one of the cases where it didn't make a lot of difference. The first irrigations 1, 2, 3, 4, 5, 6 in the post. A lot of inoculum in this one, maybe not a big deal. But in this and something on a lower level, we can see not much in 1 and 2, a lot in 3 and 4, 5 and 6, a few at the end, a few. So you get the idea that most of the inoculum is coming off in that 3 and 4 and 5 and 6 irrigations. And that's basically between a half hour to the 90-minute mark during the test. That's one parameter we need to know. The other question is how much water. Not all containers used in nurseries are the same size, so at some point we need to figure out some way to determine how much water we're going to apply to each of these. We need enough to maximize zoospore release and leaching through the bottom of the containers, but again, we don't want to apply so much that we're just diluting our inoculum out and giving a lower amount, making it harder for the test to work. So we did some tests, and where we did see in those tests that if we cut down the amount of water beyond some level, we ended up having poorer recovery of Phytophthora. And so through a number of tests, we kind of ended up standardizing at this around 17.5% on average of the nominal container volume. That's roughly about a sixth of the container volume. Whether you're working with containers of this size or very small containers, you can make those calculations, and we have these on the website. But you'll see that if we have a very small container, we're not going to apply very much volume of irrigation. Whereas if we go up to something like a number 15 container, which has got a huge volume by comparison, we have to apply a lot. In this case, we're applying 8 liters here to each pot compared to 12 milliliters down at this end. But we're keeping these in this around 17, 18 range, etc. We try to round them off so that they're even numbers instead of some odd number that you're not really going to be able to match anyway. So to apply the amount of irrigation water we want to each of many different plants that are being tested in a given test, we want to have some easy way to do this. And the easiest way is to use a known flow rate and then water each plant for the same amount of time. We can do this by measuring the flow rate put out from our irrigation nozzle and then timing it using a metronome bead. So really all we need to do this with is the graduated cylinder, some other volumetric equipment that's scaled to the size of the pots we're using. And we also need a metronome, and you can just download a metronome app, put it on your phone and use that. If we're testing a lot of different plants, and here we see that we've got several different plant sizes being tested at once in this nursery, what we want to do is figure out timings that are going to work out for each of these different plant groups so that we can organize our testing. On the website also, we've set up some metronome timings for different pot sizes, give you an idea of how many beats you would need to irrigate things at these different types of flow rates. When you're using a metronome, you aren't constrained to keep it at 60 beats per minute, which would give you a 1 second per beat. You can use intermediate timings, but we generally find that we don't really want to go much above 75 beats per minute. It's just too hectic a pace to listen to. So 90s we don't usually like to use. For a given test, what we will then do is figure out what containers we are going to use and what would be some of the combinations of beats per minute it would take to get these volumes. So if we're doing some DV40s here, we can use different types of flow rates, vary a little bit here. But if we don't have much head space, we're going to have to stay with the lowest rates. If we have a little more head space or a faster draining container, then we can push it up a little bit higher. So we've been able to take this system and try it in lots of different nurseries under lots of different types of benches and different sizes of stock. You can adapt it to all sorts of different systems. You can either use the plants in place, or you can clear some benches off and set up an area for testing. Sometimes that's much more convenient or efficient. We have, in some cases, used carts. This is a little cart with a mesh top, and we're able to use that. And in some cases, you might find that your tables that you're working on are a little bit high. That's the case here, so we had to put some spacing, some blocks underneath to raise the vessels a little higher so we wouldn't have a lot of drop between the bottom of the catchment and the vessel, because we don't want turbulence. And we have successfully baited Phytophthora ranging from 15-gallon containers, or number 15s, where we apply 8 liters of irrigation per pot. And if we're testing 6 containers, which were the most I think we did at a time for this, we're looking at 288 liters going through this vessel here. We've also done in our various studies small scale things where we're looking at other questions, but in this series of SC7 containers, we have 20 in this containers. They only get 20 mils per pot, so we're only talking about 2.4 liters for the whole irrigation. You'll see here we're using a small scale version of the zoospore collection vessel. This is our mini zoospore collection vessel. We know we're just going to have just barely over the amount that fits in a bag. we can equip a bag with the same type of drain that the standard vessel uses and work at that for these small numbers of plants. And in fact, if you know you're not even going to overrun the bag, you can just drain it directly into the bag. We've done that for even single containers. These are SC7 containers. They only get 20 mils of irrigation per each event, and so for the whole test there's only 120 mils of leachate or less, and that's not enough even to float the pears, so it's just the pears just sort of sitting in the water there. And we have had detections under all those conditions, which is kind of interesting because we were looking at total volumes of water from 120 mils to 288 liters. That's a 2,400-fold difference from the smallest to the largest. Funding for this research is provided by these entities shown on the top here, and we get support from CDFA and the Rizzo Lab at Davis, particularly for various testing, technical issues, and the Rizzo Lab for helping us secure space at Davis for some of our experiments.